To understand how the brain processes time, we start inside an MRI machine. A human subject lies completely motionless, instructed not to move a muscle. The experimental task is entirely passive. The subject wears headphones and listens to a sequence of auditory rhythmic intervals. There is no tapping, no nodding, and no physical movement required. This fMRI signal from a motionless listener shows intense activation in the basal ganglia and supplementary motor area. These signals create a biological contradiction, as neuroanatomy defines these regions strictly as motor execution centers. This raises a central question. Why does a purely auditory rhythm drive the brain's deepest movement centers when the body remains perfectly still? Finding intense motor signal in a motionless body suggests these regions are repurposed. In this context, the basal ganglia is tasked with something other than movement, computing time. For decades, medical textbooks classified the basal ganglia as the extrapyramidal motor system, a subcortical loop dedicated to the physical output of movement. Neurologists built this model primarily by observing what happens when the system breaks down. They linked basal ganglia damage to the slow, rigid akinesia of Parkinson's disease, or the involuntary movements of Huntington's disease. Modern cognitive data reveals a significant gap in that physical-only framework. When subjects perform implicit learning tests, like probabilistically classifying cards to predict the weather, these exact same deep brain structures light up. There is no motor component to a weather prediction task, yet the basal ganglia fires. This overlap between motor and cognitive tasks points to a different role. The evidence suggests the basal ganglia functions as a predictive gating engine, calculating probabilities to determine which neural programs are enabled. To understand how the brain feels a beat, we have to decode how this gating engine extracts temporal predictions from sound. In their fMRI study of the motionless listener, neuroscientists Jessica Grahn and Matthew Brett tested exact auditory variables to isolate which specific sequence structures successfully induce the internal sensation of a beat. The first requirement is an integer ratio. A rhythmic sequence built on intervals with a clean 1 to 2 to 3 to 4 relationship allows the brain to encode the sounds against a baseline mathematical pulse. Mathematical regularity alone fails to reliably trigger the basal ganglia's predictive engine. The sequences also require regular perceptual accents. The brain perceives certain notes as naturally emphasized based entirely on their temporal grouping, like a note that follows a long pause, even if the actual audio volume never changes. This schematic shows the tested stimuli. The top timeline, metric simple, successfully induces a beat. It combines clean integer ratios with regular spacing of perceptual accents. Compare that to the middle timeline, with irregular accents, or the bottom timeline. Neither reliably induces a beat. The basal ganglia remains quiet during irregular sequences. It activates only when the incoming auditory signal provides both a mathematical ratio and a regular accent, allowing it to lock onto a predictable temporal grid. To understand how this grid pushes the brain toward action, we must trace the neuroanatomical wiring connecting the auditory signal to the motor network. The direct pathway starts when the cortex sends a positive 1 signal to the striatum. The striatum fires a negative 1 signal to the GP int. Normally, the GP int continuously sends inhibitory signals to the thalamus, locking down the motor cortex. When the striatum fires, it inhibits the GP int. By inhibiting an inhibitor, the direct pathway creates a mathematical double negative. With the inhibition lifted, the thalamus is free to fire a positive 1 signal up to the motor cortex, enabling a specific motor program. The direct pathway acts as a precise predictive release valve. When the brain anticipates the next regular interval in a rhythmic sequence, it triggers this double negative loop to release the internal perception of the beat. To successfully enable one target program, the brain must suppress conflicting programs via the indirect pathway. Striatal neurons send a negative 1 signal to the GP external, which normally inhibits the subthalamic nucleus. By inhibiting the external segment, the subthalamic nucleus is disinhibited. It fires a positive 1 signal to the GP int. This triple negative calculation causes the GP int to clamp down even harder on the thalamus, locking out competing programs. To manage both pathways at once, the system relies on a master tuning chemical, dopamine, arriving via the nigrostriatal projection from the substantia nigra. Dopamine hits both pathways simultaneously, but with opposite effects. Binding to D1 receptors, it excites the direct pathway. Binding to D2 receptors, it inhibits the indirect pathway. This dual action tips the total neural balance toward enabling the targeted action. Beat perception requires flawless synchronization. Dopamine tunes the network to release the target temporal beat while actively clamping down on all asynchronous auditory noise. Applying this neuroanatomical model to our motionless listener resolves the paradox of the active MRI scan. When listening to a metric simple rhythm, the evenly spaced accents provide a clear predictive cue. That cue is recognized by the brain and fed into the striatum. The intense, bold signal we see lighting up the putamen perfectly correlates with the active firing of the direct pathway. The striatum is actively disinhibiting the GP-int on exactly the predicted interval. The motor plan to physically tap a foot or nod a head to that beat is fully calculated and queued up in the basal ganglia, even though the primary motor cortex never executes the final command to move the muscle. The brain simulates movement to the rhythm, computing temporal probabilities through its deepest motor circuits. This neurobiological precision raises an obvious question. Does a temporal prediction engine this complex require years of specialized musical training, or is it innate? This bar graph compares proportional fMRI activation in professionally trained musicians versus untrained non-musicians during rhythm discrimination tasks. Looking at the right side of the graph, we see massive activation spikes in the cerebellar and premotor regions for the musicians. Professional musicians rely on active rehearsal strategies, recruiting immense processing power from the cerebellum. But looking at the far left columns representing the supplementary motor area, the statistical difference between a concert pianist and an untrained listener disappears. The activation is nearly identical. Forming a temporal representation in relation to a beat operates identically in all human brains. It does not require practice or expertise. The basal ganglia functions as a hardwired probability and timing computer, integral to how we perceive the world's temporal structure. The human motor system performs continuous temporal prediction. We perceive the passage of time through the very circuits that prepare us to act.